Good morning, everybody. Thank you for joining us to this webinar, which is part of a series that we're happy to offer to you about the Blue Cloud Virtual Lab. I am Sara Pittonet. I am Senior Project Manager at Trust IT, and I have the pleasure and honor to be the coordinator of the Blue Cloud initiative. Let me first introduce to our newcomers today, Blue Cloud. Blue Cloud is the Future of the Seas and Oceans flagship initiative of the European Union Horizon 2020 program. And it is the thematic European Open Science Cloud for the marine domain. It aims to promote the sharing of data processes and research fundings in the marine domain by delivering a collaborative web-based environment where researchers can access an unprecedented wealth of marine data resources and interoperable added value services and products. The project started in 2019, features 20 partners and 13 Blue federated infrastructures that make the data available to the users. What's strategic in this architecture is exactly the federation approach. Blue Cloud federates leading European cross-disciplinary infrastructures, namely EUDAT, D4Science, Copernicus DS, Wikio, with 13 long-term data management infrastructure. And altogether, we have built the Blue Cloud technical framework, where scientists can access the ocean data, the tools and the services and all the research outputs there to perform the research in a more efficient way. As part of this framework, there are 5 virtual labs that have been developed as real-life demonstrators, making use of these resources, driving the further development of additional features. They address specific challenges tackled by researchers in 5 domains: biodiversity, genomics, environment, fisheries, and aquaculture. Today, we are going to have an overview of the demonstrator on Plankton Genomics, which is led by the European Bioinformatics Institute and created by the Faculty of Sciences at Sorbonne Université with the contribution of the Flanders Marine Institute. The virtual lab has developed 2 services. One, a notebook which is exploring genetic data and defined clusters containing unknown genes, and another service which allows to map the geographic distribution of plankton functional gene cluster using habitat prediction models. These are available in the virtual lab, and our colleagues today are going to show you how they can be used and further exploited by researchers. We hope that the webinar today will inspire new users, will inspire you, as happened during the Blue Cloud Hackathon that we have organized in February this year, where almost 150 participants came from a very diverse community of marine science practitioners, computer science, and innovators to test the Blue Cloud web-based ecosystems and perform their marine research. I'll leave with that, and I'm very happy to give the floor to Eric Pelletier from SIA, who will start the overview of the virtual lab.
Thanks a lot, Sarah. Okay, now I can share my screen. Can you see it?
Yes.
Okay. So I will just give you an introduction of the data that we are using in there. So as you said, it's Plankton Genomics, it's eukaryotes. We forgot to mention that for the data that we use in this demonstrator is only eukaryotes data that came from a sampling that was performed 10 years ago during the Terra Ocean campaign. And so this is a campaign that sampled more than 200 places around the world in the different oceans. and we aim to try to describe and understand what are the plankton organisms that live in the upper part of the ocean, in the photic zone. So this is the part in which the light can access. So from surface down to something about 150 meters depth. And we mostly use the omics approaches, so the sequencing of DNA and RNA, to try to describe those organisms and to try to understand their functions and how they can operate with their environment. So this is the data that is in the core of what is available on the demonstrator. So in order to access the organisms, we perform assemblies and we define what we call MAGs. So MAGs are meta-genome assembled genomes. And we define them by co-assembling a collection of samples. So we define 11 set of samples that are depicted there in the different colors. So we co-assembled all the sequencing reads that come from a set of stations, and then we perform an automatic and manual binning to construct contigs that we try to assign to a given organism. So I won't go much into detail in the process. It is quite well known, and we recently published the paper last week that described all those data. So you can find it on Cell Genomics from Tom Delmont et al. Last week. So that describe all this resource. But basically, what we do is we co-assemble reads that come from a collection of places. We define contigs, and on those contigs we define genes. And we also use the abundances of those contigs in the different samples that we define by mapping the metagenomic reads on the contigs to perform a binning. So the idea is we put together the contigs that have the same profile of abundances in the different samples. So this is a key point. Rather than assembling point by point, we co-assemble dozens of samples at the same time, and then we use the abundance profile to cluster together the contigs that are supposed to belong to the same organisms. And then we perform a manual curation that allows to have a high quality set of MAGs that are representative of plankton organisms that live in the water that we sample. So this is the first step, is to construct genomes and to use these as the starting point for the analysis, the subsequent analysis. So once we have those contigs, so the first thing that we did is a phylogenetic analysis to describe what are the part of the tree of life that are covered by those MAGs. So we defined a 714 different MAGs, and this is a picture that depicts the phylogenetic distribution of those MAGs, and that we allowed to cover the main part of the tree of life for the plankton. So the dinoflagellate, the ciliates, the diatoms, and so forth. I won't name them all, but this is a figure to show that we cover the most part of what is known to be there in the ocean. So what are the data that we used in this demonstrator? As I said, we start from the MAGs. For each of MAG, we have a taxonomic assignation. From each of the MAG, we also define genes, and those genes were support for different kind of analysis. The first one is to perform a functional annotation, so to try to give a function to those genes, which works in only half part of them. So 50% of the genes, we don't have functional information so far. What we perform also is the sequence comparison of the protein sequence of those genes to define protein clusters that will be the second important part of that data that we use for the analysis. So these protein clusters will be analyzed based on the different way, the functions and the sequence comparison. What we did also, we use the metagenomic reads to define the abundances of all the mags in all the samples. So we have amount of DNA in the different samples, and we use the metatranscriptomic data, so the meta-T reads, to define the expression of each of those genes in each of the different samples. So we have the abundances of the mags and the expression of the genes in all the stations. So what we did then, so as I said, from the mags, we defined a set of proteins for which we have known or unknown functions. We clustered them based on the sequence similarities. So this is called the sequence similarity networks. So we define different protein functional clusters that associate the idea is proteins that are similar in terms of sequence probably bear the same functions. So what is a sequence similarity network? Is a collection of proteins. So each node is a protein. The proteins are connected based on the sequence similarity that we define. So we basically have percentage of identity between different proteins, and each protein is associated with the collection of descriptors, so the taxonomy, the function, and the abundances and the expression of those genes, of those proteins in the different samples. So this is the basic elements that are used as input for this for the tool that Alexandre will explain a little bit more later. So this is important to have an idea of what are the data that are in there. So it's eukaryotic. It's about 10 million genes, so 10 million proteins that are defined from those 713 mags. As I said earlier, we have, I didn't put it. So we have 713 mags that are eukaryotic, that cover the large part of the tree of life. We predicted 10 million genes from those mags. We have the annotations for the functions and the taxonomy of each of those proteins, and we have the abundance and expression profile of those genes in the different samples. So this is the starting point of the data. Thank you.
Many thanks, Eric. I take just the chance to remind that you can use the question and answer if you have any questions. And I leave the floor to Jean-Olivier Irisson now from the Sorbonne Université.
Yes, so hello everyone. So what I'm going to present now is so Eric presented the data. I'm going to present the theory on the computation that is done, and then you'll have the actual application and the results presented by Alexandre. So as Eric presented, what we start from is a metagenomic signal that comes from discrete places in the ocean. And even though the Tara sampling was quite extensive, it's still just a few points in the ocean. And the idea, if that works, yes, is that if we have a few points in the ocean like this, with some quantitative information, what we would like to get to is a complete map of the abundance of our variable of interest. So in our case, the target variable would be the abundance of a certain gene, therefore a certain function in a certain organism. Well, actually, it won't be a gene, it would be a cluster of genes, as Eric presented. So how do we go from a few points to a complete map? Well, that's one way of doing it is the principle of habitat modeling. So what we say is that we assume that there's a relationship between the abundance of the gene cluster that we're trying to predict and the environmental information, so the environmental conditions. So and the environmental conditions, we have resources that give us complete maps over the whole world of these environmental conditions. So here I'm presenting an example with just one environmental variable, which is temperature, but often, I mean, we don't do it with just one, we do it with several variables. So the idea is that for each point, we'll fetch the temperature information, and we'll then plot the abundance signal that we get from the point with the temperature data that we get from the point. And through this, we define a response curve of the abundance of our target variable according to temperature. Okay, that response curve is called, in the case of species, it's called the niche of the species. Here we're talking about gene clusters, so we don't really talk about the niche of gene clusters, but it's a response to the environment. Okay, so we do this from several points to construct that curve. And then once we have the curve, then it means that we can take any point on Earth, even and especially a point where we don't have any observation. Then we can look at what the temperature was here, and then we can predict what abundance we expect in that particular pixel. And we do this for every single pixel of the world, and then we have a full picture of the abundance of our target variable across the world's ocean. Okay, so it's not exactly the abundance of the target variable, it's the potential abundance. It's the regions where we think the environment is favorable for that particular target to be abundant. Okay, so that's the general principle of habitat modeling, and I'll get into a bit more detail about how we apply this to the metagenomic signal from Tara oceans. So the first question is, how do we construct that response curve? Okay. To construct that response curve, we chose to use regression trees, which is a statistical technique that has the good idea of being quite efficient and quite practical to use. So what do we need to construct a regression tree? We need the response variable. Okay, and here I'm presenting a simpler example with, sorry, with just presence and absence. Then we need a metric to evaluate whether our tree is working or not. And so in the case of presence and absence, it's quite easy. You just say, okay, the tree is predicting that my species, let's say, will be present. Is it present or not? Okay, and we compute the percentage of correct predictions. And we need predictor variables. And in our case, the predictor variables are environmental variables. So let's say we start with 200 stations, which is the number of stations in Tara approximately. So we have 200 observations of presence and absence, and then we have 200 values of temperature, of salinity, of silicate concentration, etc. And we're trying to separate these observations into small bags, okay, according to the value of environmental variables. And if we do this, we see that we would separate the places where the temperature was warmer than 8.5 degrees on one side and so warmer on that side, sorry, and colder on that side. And that would separate regions where the species is mostly present from the regions where the species is mostly absent. Okay, and what we do is that we evaluate that proportion of presence and absence on either side. Okay, so the question, so we have this tree that separates our data in small bags. We can evaluate how pure the bags are. And the question is, how do we find the best split? Okay, we have many values of temperature, we have many values of salinity, of silicate concentration, etc. How do we choose the best one? Well, it's quite simple, actually. You try everything and you see what's best. And that's what it's called machine learning, because you just let the machine do all the work. So that's a simple regression tree, okay? And in our case, what we have is not one concentration, we have several concentrations or several abundances of several genes, okay? And it's really important to consider them together, because the genomic signal gives us good information on the different proportions, but it does not yet give us an information on the actual numbers, okay? So what we have as information is who is dominating in the ecosystem, not exactly how many there were. So because it's an information on who is dominating, the signal is really not in the value here, but in the proportion. And that's what we're modeling, okay? So for each one, we would have, so our response variable would be a vector of concentration, or standardized reads in our case, which I will call Y. Okay, we'll make a prediction, and in statistics we call the prediction with a small hat. Okay, so Y hat. And then we need a metric to say how pure our bags are, and our metric is the multiple root mean square error, which is the difference between the two squared, then square root, and we sum this for every cluster of gene. Okay, so it's basically a simple regression. It's the same metric as in the simple regression, but no, it's multiple because we have multiple clusters of genes. Finally, our explanatory variables in our case would be climatologies, so they would be long-term averages. And the reason that the long-term averages is because we're looking at metagenomic signals, so we're looking at who is there. Okay. And who is there is really a result of the regional long-term conditions. Okay, we're not looking at exactly, that particular date in time. We're looking at what is the community looking like, and that is shaped by the average temperature and the average salinity rather than a precise salinity and temperature at a precise point in time. Okay, so we have our three ingredients. We can fit the trees. Okay. Then I'll go into a bit more technical details so that Alexandre does not have to. So we don't actually do regression trees, which is just one big tree on the left. We do what's called boosted regression trees. So instead of building one tree that fits the data as best as possible, we build a small first tree that explains some of the data. And then we see what we don't explain, which is called the residuals, and we fit another tree to that. And then we take that and we take the residuals from that and we fit another tree, et cetera, et cetera. And we fit this way hundreds to thousands of small trees as opposed to fitting just one big one. The reason we do this is simply because it performs better and it gives smoother response curves. So for habitat modeling, it's really more appropriate than having a single big tree. So the principle again is we have the response variable, we have our explanatory variables, okay, temperature, salinity, oxygen. Okay, we have the association in the points that where those are observed. We fit the model, we have a prediction, we compute the error, and we fit again until we have something good. Okay. Once we have this, we can take the temperature, salinity, oxygen in every place of the world, make it go through the model, and predict our response variables. No, we're not predicting just one thing, we're predicting several things. Okay, and those are the maps, the complete maps. Okay, what we do in addition is that in that box, in that mysterious M box, there's actually a few parameters that we need to define. Okay, we need to define how many trees we do. We need to define the depths of each tree, so how complex the small trees are, etc., etc. And we can't do that on just a training set because otherwise we would fall into something that's called overfitting, meaning that our model will be really good for our data sets, but really crap for everywhere else. Okay. So to avoid this, what we do is we do what's called cross-validation. So we take our observed data, so the Tara stations, we cut that into several pieces. Here I've cut it into 3 pieces, okay? And then we fit the model on the first 2 pieces, and we try to see how good it does on the remaining piece. So because it's an independent data set, we're not fitting our model precisely on the data here, the blue and the red. We're fitting it in a way that it still predicts data that it has never seen, okay? And we do this on the blue and red to predict the green. We do then the red and green to predict the blue, and the blue and green to predict the red. Okay. This way, we can find a model that is both really able with the best setting to predict our data, but still that can generalize to other data, which is what we want. Finally, we don't want to just have one map of prediction. Okay. It's also really important to estimate how confident we can be in our prediction. So we don't just want the Y hats, okay? We don't just want a prediction. We want also a sigma, so a variance in that prediction. The way we do this is we do this by bootstrapping. So what is bootstrapping? We start from observed data. Okay, we have the 200 Tara stations, in my example, just 6. Okay, and we fit the model the way that I explained before. And then what we do is that we create a new fake data set where we just resample the data that we have. So we change the order, but also we repeat some stations and some others are missing. Okay, like station 2 is there 2 times, station 1 is there 2 times, but station 4 and 5 are missing. Okay, and we fit a new model that will be slightly different from the first one, but still realistic. It's just a different, starting point. And we do that several times. So that means that instead of having just one model, we have many models. And then these many models, we can apply them to the whole world. And that means that we get many predictions. And these many predictions, we can compute the average of those. That would be the map, okay, where we think that particular gene cluster will be abundant. But we can also compute the variance between those, and that will give us an idea of the uncertainty, okay? So in the end, to exploit the genomic data that Eric showed, what we do, if we spell it completely, will be bootstrapped, cross-validated, boosted regression trees. So that's a lot of complicated words, just to say that we're using the environment to predict maps, and we're getting uncertainty together with those maps. Okay, and with that, I thank you and leave the floor to Alexandre.
Many thanks, Jean-Olivier. We had a couple of questions. Not sure if you want to have a look at them now, or if maybe
as you wish. I think we have time.
Yeah. So let me open them. Yes. So one was already addressed by Eric.
Oh yeah, there's a chat and the question and answers, or?
Yes, so we had one question about the prediction of the abundance of plankton. What I understood, the data from Tara punctual simplings, not repeated along annual fluctuation season, and the opposite to predict the temperature related abundance. You're using the average temperature. So Eric replied, and if you want just to comment wide.
Okay, well, I think.
Eric, yeah, or Jean-Olivier or Eric, but anyhow, it was just to comment on this. Then we have another question waiting.
I think so. Of course, there's tremendous variation in time, in addition to variation in space. That's a difficulty, I would say, of the Tara Oceans data, because the ship is moving, so it was at different places at different times. So we have what's called an aliasing between time and space. Okay, we can't know whether it's time or space, and indeed the only way to know is to set one. So stay in a fixed point and go through time. That's what Eric answered. It's you need to have time series to resolve this. But another response in the specific case of the work that we did is that, as I explained, we're using only the metagenomic signal. So Eric presented the fact that there's metagenomic and the metatranscriptomic. The metagenomic is what are the genes that are there. The metatranscriptomic is what are the genes that are expressed while Tara was there. And clearly for the metatranscriptomic, it's the local immediate conditions that influence which genes are expressed. Okay, so it would not make sense to relate the transcriptomic to long-term averages, because it's immediate expression. But for the metagenomic is which species were there, basically, and what genes they were carrying. And that is shaped not by the immediate condition at the time that Tara was there, but by the overall regional conditions over the long term. What evolutionary conditions are favorable to one type of community versus the other. And because this is the data that we're looking at, the genomic, so the long-term, community structure, then it's more understandable to use long-term climatologies. So yes, temperature varies in time, but depending on your question, you should consider or not consider that variation. And in our case, we don't consider it.
Thanks. And then we have a question by Ivan. On the modeling, isn't there seasonal variations not taken into account? For instance, extrapolating from data taken in South Pacific, defining habitats to areas in North Atlantic, even with the same temperature. relation to nutrients for currents differences?
So I would say the answer would be the same. Then it's with an additional point maybe is that indeed one hypothesis of habitat modeling is that the community is shaped by the environmental conditions. And it does not matter whether you're in the Northern Atlantic or Southern Atlantic or Southern Pacific. If the conditions are the same, then you would predict the community to be the same. Okay, that's an approximation and a hypothesis because it's not true in an immediate fashion. Okay, that means that the model will never explain 100% of the variance. And as we will see, it does not explain 100% by far. But it's, you suppose that some of that community structure is driven by the environment, and that's what you're trying to predict. And then, regarding the seasonal variations, yes, there's seasonal variations, but again, because we're looking at long-term community structure, that's not what we're trying to capture.
Just to add a comment on the season aspect, just to keep in mind that most of the sampling was performed during what we call the summer-like time. So we planned the trip to be more or less during the 6 months of the summer in each of the stations, even in the Arctic. We can go a step further, saying that we only sampled at day, not in night. So there is the time, the daily aspects that are not captured by this data, but it's very complicated to go there. So to set up stations, long-term stations on open ocean, it's very difficult. So far, most of the time, series are performed in coastal waters because it's easy to go there every week or every fortnight or every month. If you want to go in the middle of the Pacific Ocean every week, it's very, very complicated. So it's difficult to have long-term time series in open ocean. But there are some automatic devices that are being designed, and we start to build some of them to try to have sampling performed on those locations. But it's very, very complex because you have to keep the samples in conditions that allow to do genomics beside. So it's very complex in the next decade, maybe.
Thanks, Eric. So I would suggest to move on now with the Alexandre's presentation, and then we go back to Lucia, who also has a question.
Thank you, Sara. So I'm Alexandre Schickele, postdoc at Sorbonne University, and today I will introduce you more the Plankton Genomics virtual lab, how you can access it, and further applications or development that you can do with it or that could have been done during the hackathon, for example. So first I will talk about the access to these open resources. So the virtual lab, the video tutorials that you have and all the documentations, as well as the data you can find on Data for Science. Then I will present you an application on C4 carbon sequestration metabolism that is based on the virtual lab, but also includes further developments. So first, about the resource accesses. When you arrive in the Blue Cloud D4 Science page, you will have this pictogram here that will bring you to all the demonstrators that are available. You click on Plankton Genomics Demonstrator 2, and you will end up here in Plankton Genomics with 2 notebooks. If you want a live demo of all this, you have video tutorials on the Blue Cloud YouTube channel with 10 and 20 minutes formats that were done for the hackathon and demonstrator tutorials. So in the Plankton Genomic virtual lab in D4 Science, you have notebook 1 that is about what Eric presented before. So the Marine Atlas of Tara-Ocean unigenes, as well as all the construction of the network of sequence similarity of protein clusters. And you have notebook 2 that is about projecting plankton functional gene clusters. So what Jean-Olivier introduced you, and with all the codes and documents accessible through D4Science. And I will focus more on this notebook 2 as it's the application part of this virtual lab. So when you click on notebook 2, the content, you have a big zip file here that can be used in Data4Science or for a personal installation if you want to have further development on it. It contains all the code, all the documentation that I will explain you later. And you have also 2 Word documents with how to access the data, how to install the notebook, and the user tutorial as well. So you should have all the information for using it here. When we open the notebook in Data4Science, you will have here, it will look like this. So with all the notebook files, the data, all the codes, and Python notebook, which is documented with all the modeling steps of this notebook. So a description, the code that is embedded in the notebook, and directly the output. So you can use it directly with this Python notebook. Or if you want to do further development, you can always take the code directly here and do whatever improvements you need. Concerning the data available, so we don't have all the data of Tara Ocean in the notebook 2. We only have the data for pico eukaryotes, so 0.8 to 5 micrometer size, surface sample only, and this is about 102 Tara Ocean stations. And we have species like Micromonas, for example, that are included in this data set. Sorry. The genomic data are metagenomic reads normalized per kilobase, so by the size of the gene. All genes are functionally and taxonomically annotated at different levels and grouped in connected components, so the clusters that were mentioned before. Here in this data set, it's clusters of 80% similarity and 80% identity. From this big data from 102 stations and 4 million genes, or 1.2 million connected components that were found in pico eukaryotes, we can select genes or connected components that are related to a function or a pathway of interest, let's say carbon sequestration, for example. All the genes that would be related to carbon sequestrations are still too big to be modeled by boosted regression trees, because we cannot model 500 genes with only 100 stations. So we need to do a dimensional reduction that is implemented in the notebook by abundance, for example, or spatial coverage. So we would like to model the genes that are related to carbon sequestration and widely distributed, or the rare genes, or the same with connected components. And we end up with about 50 connected component or genes, depending on what the user selected. So with these 50 connected component or genes, we have a table of 100 stations and the relative abundancy of our 50 genes or connected components, as well as the environmental values from environmental climatologies for the same 102 stations. Very quickly, this goes into multivariate boosted tree regressor. So what Jean-Olivier introduced you before, with n-fold cross-validation procedure that can be user-defined and the tree is fitted using a mean square error loss function to optimize the tree fitting. The user has access to user-defined parameters, so the learning rate, how fast will the different sequential tree learn from the residuals of the previous one, the size of the tree leaves, the depth of the tree, etc. You have different parameters. And once you have fitted a model, it will be evaluated using multivariate root mean square error and R squared. The plots of the observed against predicted values are also displayed in the notebook, as well as, if you wish, the importance of each environmental variable in constructing the tree. Once we are happy with the model, we can do projections by genes or by connected component, depending on which modeling level we chose, according to a bootstrap method that will predict us n bootstrap map that we can then average into one map per gene or connected components, and that includes the uncertainty. So here the color scale is the abundancy, and more it's darker it is, more you have a coefficient of variation associated with your projections. So you can see where you have a projection that is likely to be more precise and how abundant your gene is at this location. All these projections can then be aggregated according to their functional or taxonomic annotation of the gene or the connected component. So that was for the virtual lab, and we applied this. So here I will show you an example on C4 carbon concentration mechanism. That is a widely distributed pathway, especially as well as taxonomically in pico-eukaryotes. And it also includes parallel pathway within this carbon concentration mechanism, which is very useful because we are working with relative abundance. So very quickly, photosynthesis happens with the Rubisco enzyme that can fix either CO2 or oxygen. And the problem is that it has more affinity towards oxygen, so it's more likely to do photorespiration instead of photosynthesis. To counter that, there is pathways that will concentrate the CO2 in front of the Rubisco to allow the cell to be more likely to do photosynthesis and not photorespiration, which is a wasteful process for the cell. So here we have our data sets of C4 related connected components that were selected among all the connected components that are available in the virtual lab. We sum all the lines to one to have our relative abundancy, and then we do dimensional reduction. So this is a further development that we needed for carbon sequestration because we have many genes. So basically we will select the 50 genes whose pattern are representing the most of the variance of the whole data set. So we selected here 50 connected components that represents 95% of the variance, and this is our data set that we will enter into the model. We do our modeling as explained before, and we will have projected relative abundance for these 50 connected components. All projected relative abundance were standardized to 1. I will come back later to that. Now we have also 200 connected components that we didn't select for the model, but that have a pattern maybe that is close to the pattern of a connected component that we selected. So for that, we did in the component analysis space distance calculation to see which of the here 200 not selected connected components, which selected pattern is the nearest for each of them. And this allows us to rebuild the projections for these 200 connected components that were not selected according to the projected maps of the nearest pattern. So here we have outputs then that can be aggregated taxonomically or functionally, and that are all standardized. So the average, let's say, would be the average pattern of each enzymes in the pathway. But we can also rescale these data by their proportions in the original data set. So include the dominance of maybe one connected component over the other, and by extension of one taxonomic, taxa over the other. So here with the rescaled data, which would be more the realized distribution instead of here for the potential, we have here a taxonomic, we include potential taxonomic effects. If we look at the map, so here you have again the metabolic pathway with the three parallel pathways within carbon concentration. So you can see that all the maps look quite the same. So here's the potential projections, so not rescaled by the proportions in the raw data. We can see that we have the highest potential abundancy is always in tropical waters. So maybe we have more, we're more likely to have a higher abundance of each of these enzymes in these areas. If we look at the correlations, all the enzymes of this pathway are quite well correlated between them. And if we correlate each of the enzymes with the maps, so we can construct taxonomical maps as well. And if we correlate the taxonomical map with the functional map, we can see that here we have a very low taxonomic effect, just maybe PEPDK, that is an enzyme that is a little bit different in its taxonomic composition than the others. Now we can also rescale all our projections by the who dominates in the raw data. And this gives us a result that is slightly different, so which would be more the realized abundancy, relative abundancy of each of these enzymes. Here we can see that more or less all enzymes are have higher abundance in the tropical water, but Schickele here have its higher abundance in the Southern Ocean. And we can also see difference here in the rich Pacific waters, between two enzymes that would be in the same pathway. So for example, here you have a maximum abundance, and this goes to next to this enzyme that has its minimum. So this is a bit strange, but when you look at the correlations, so as I said, you have PEPDK that is very different from the others, and some non-correlated enzymes. When you look at the correlations between the functional maps and the taxonomical maps, here we can see a taxonomic effect. So maybe the discrepancies that we had here are due to a taxonomic effect between the different sampling locations, or that because we don't have an exhaustive sampling, maybe that some enzymes were not sampled in all species. So here you have the difference, three functional groups, sorry, and three taxonomic groups. If we look a bit overall, we have C4 carbon concentration mechanisms that have a high functional redundancy in tropical ocean with different pattern, especially in the Pacific, between nutrient-rich and oligotrophic water, and also important differences for some enzymes between realized and potential abundances. This can be due to non-exhaustive sampling, especially in the polar region, where we have a very few sample, especially in the Southern Ocean. And we may, because we have very few sample, we may have a dominance of one species in the sample that is annotated, a gene that is annotated to one species and one function. And then when we project it, we extrapolate this dominance, which may not be totally true. So that's why we have a strong taxonomic effect on the realized distribution. To explain that, we need to check the variable importance. So in our model, we had chlorophyll, euphytic zone, and nutrient-related variable that were very important in constructing the model, but we are still trying to implement yet the average functional response to each environmental variable, so to better understand the distribution and maybe a relation to metabolism. Are some enzymes present here because of nutrient concentration? Yes, but in rich areas or in poor areas, and trying to differentiate the effect of each variable on each functional map. And then for now, I only talked about connected components, but as Eric said before, there is a lot of genes. So a connected component contains several genes, and there is a lot of genes of the network that were not functionally or taxonomically annotated. And we can use this virtual lab at the connected component level, so modeling the different genes that are in the same connected component to see if there is a relation between unknown genes and annotated genes. And overall, this virtual lab is an open science tool with wide applications. I tried to show you the first results of one type of application that you can do with it, but can be applied to a diversity of metabolic pathways that can be related to biochemical cycles, such as carbon sequestration, or maybe some metabolite synthesis, and it's here for you to use it. Thank you very much for your attention.
Many thanks, Alexandre. Thank you all. So I saw there was also a pretty active chat. So maybe, do we need to go back to Lucia's questions? I believe it has been addressed already. So I'm not sure if there is any need to go back on this, Eric. Otherwise, yeah.
Yeah, I can comment a bit because that's a very good point, because we know that we only scratch the surface of the organisms that in there using the MAGS approach. As I said before, it's only from 10 to 40% of the metagenomic reads that are captured by the MAGS. So there is a large amount of organisms there that are not depicted. When we use barcoding approaches, so based on 18S or 16S, for the prokaryotes, we can go much deeper into the representation of the organisms that are in there, but for which we don't have the genes or part of genomes to study the function. But even though we can look at the composition of the communities, and then we can tag the barcodes that are associated with the MAGs to try to define are the MAGs marker of the presence absence of other organisms. The key point there is that in the MAGs we don't have the 18S or the 16S because of the assembly process. Those genes are too conserved to be assembled separately for the different mags. So we don't have connection between the 16S or 18S tags on one side and the mags on the other side. And for many parts of the tree of life, we don't have enough representative to be able to connect the precise 18S with the corresponding mag. So it's very difficult to connect these. This is work in progress right now, but we don't have ways to answer this in the actual dataset that are in there.
Thanks. So before you, no, sorry, there is another question. No, it's just a thank you. Okay. So before we leave you, since we were very pleased with the interest we received in this webinar, we had more than 30 registered institutes among oceanographic centers, zoological stations, universities in Europe, but also beyond. So we got quite interest from all the Mediterranean region, I would say, Tunisia and Nigeria as well. No, Nigeria, no, sorry, it probably is wrong, but Algeria. I would like to ask you just 3 questions. We have a Menti. Thanks, Federico. We have a Menti poll, really simply 3 questions. We'd like to know where do you come from, which is your institute, and if you might be interested in a follow-up dedicated meeting with the Blue Cloud colleagues to understand more about the features, to see if any custom tailored adaptation can be done, or how you can, I don't know, use it or further explore it. So feel free to answer that, and we'll then get back to you and organize something dedicated. It's nice. Yeah, we have South Africa as well. I saw that. Trieste. Thanks. I think you can move on maybe to the next one, Fede. We have the list, of course, of entities, since you've all provided this information while registering, but it can be nice also to have this overview for the others attending. We also have public national agencies. I saw that. Great. And then, yeah, we have a third one exactly. We wanted to understand whether a follow-up might be useful. We can have something with your team or with even a few group of people. You can extend it to colleagues and or the entire institutes. Yes, that's nice. So I think in the next question, we'll probably ask you the name. You don't need to show that, Fede, but I mean, we then have all your details, so we'll get back to you. Yes, or contact us with an email. It will be fine as well. Okay. We hope it was useful. Many thanks to our speakers today. So you should be able to find all the links and information from the Blue Cloud website and then enter in the virtual lab. If you're not signed, feel free to ask for an account. So the colleagues will moderate your registration into the platform. And we hope we'll get in touch with you very soon. Thank you very much, all of you, for this webinar today, and have a nice weekend.